Hi MS Translators, welcome to Day 2 live from the 9th Joint ECTRIMS-ACTRIMS Conference here in Milan in Italy. This is going to be our Day 2 summary of the highlights we've heard from the conference today. You know me by now, my name is Drummond, one of the co-founders of MStranslate, and I am joined today by
Travis Stiles, CEO and founder of Novoron Bioscience,
and Joanna Cobbin, member of MStranslate.
And so with the three of us here, we've gone across a number of different sessions today. I'm going to start by really thanking people flanking me for being enormous support at the conference and being able to go to all of these sessions so that we can report back to you. It's a really hard thing to be going across so many sessions, so to have them here being able to sit in and report back to you is really invaluable. So we're going to start with Jo, who's going to talk to us about a session that she was in and a specific presentation from that session that she found really interesting.
Yeah, so I went to the gut microbiome session today, and what I found most interesting was a talk from Francisco Quintana from Harvard in the US, and he was talking about the gut microbiome and regulatory pathways that he's investigated. So he was looking at a receptor which he refers to as AHR. So it's like a steroid receptor, and when it's activated, it results in the inhibition of inflammation and therefore in MS, reduction in disease. And in MS, it has been shown that there is a lower level of antagonists or small molecules that actually activate that receptor. So what was really interesting that actually came out of his talk is not only that he was able to show this, it's that he was testing what he referred to as a synthetic probiotic. So it was a probiotic where they've actually inserted in a small gene sequence which actually transcribes proteins which actually are antagonists to the AHR receptor, and then which then circulate and they suppress inflammation. So what they showed in a mouse model is that when these small molecules are actually produced by these probiotics that are taken orally, they actually reduce the amount of inflammation, and it limits the amount of inflammation in those mice, and therefore you get less animal model disease, or EAE, which is a well-known MS mouse model. Yeah. And it was really interesting. And this probiotic has actually been tested already in phase 2 trials in humans. So it's actually been shown to be safe in humans. So hopefully it's something that could be used in the future for potentially work in humans with these small molecules.
Okay, fantastic. I mean, we know that the gut microbiome is a topic that's really interesting to people living with MS. It's certainly something that's really come along in the first 5 years, and I think that's a really interesting sort of show of how sort of research topics can go from something that's interesting through to basic research now to already be something that's in trials in quite a short period of time. So it's really interesting.
Yeah,
thanks for
really
that. Travis, what was your presentation of interest today? I'm sure there's going to be more than one.
So I kind of had two main focuses today. There was the kind of what's new in like the biomarker world, which is really interesting to me because like when you're looking ahead to like potentially if you do have a new therapeutic, like what are the clinical trial endpoints you're going to be looking at. I think it's pretty well known that like MS kind of struggles with like the quality of endpoints that we have to work with, and we've made a lot of advancements, but anything that's innovative in that arena is important. And then kind of like the future of the therapeutic landscape, that's kind of been a theme of what I've tried to look at. I thought there was a lot of stuff on BTK inhibitors. There was a lot of stuff on like cladribine and a lot of like there was kind of a recurring theme. There wasn't in the presentations themselves. There wasn't like a lot of like really novel stuff. The couple things that really stood out to me is there was a conversation about kind of like a modified CAR T therapy with like an inducible payload expression system, and I thought that it was a really clever potential novel approach to the treatment of MS.
So sorry to stop you for a second. Can you explain to people watching may not know much about CAR T and how that works. Can you explain a little bit about
I'm not an immunologist, but so I'm going to give you like probably the super oversimplified and possibly wrong explanation, but it's basically reprogramming your own immune cells to attack things of interest. And so in this case, it would be reprogramming immune cells so that they could actually release a payload, as opposed to kind of, in the case of CAR T, it's really about killing cancer cells, but in this sense, it would be honing in on cells of interest and then delivering a drug, which is really potentially really cool if that works. And on the biomarker side, I was actually, there's a lot, everything's about neurofilament light chain and like serum or various forms of GFAP. The couple things that I actually thought were more interesting is there's, I think rheumatoid arthritis is a really good example of a disease where there's not one test. You have C-reactive protein, you have cytokine panels, you've got all these things, and it's really about the kind of the broad picture. There isn't one test that's like, that's what we look to. And I think people are getting more comfortable with the idea of like, well, here's the 5 things we're gonna look at, and if you have this clustering, that's gonna be good enough. And I think that that's a fine direction to go. But one of the things I was really struck by is that there's starting to be more work done in like lipidomics. And so I actually think this might be interesting to kind of explain a little bit more to your audience, because most of what we do in like drug discovery or biomarker discovery is in proteins. But proteins are only one of the big macromolecules in your body. You also have lipids, fats, and carbohydrates and sugars. But because of the technology that we use for life science kind of discovery efforts it's really DNA and protein centric. There's much less work done in this kind of like glycobiology and lipidomic type space. And so I saw some really interesting things where people were really starting to stratify and mainstream the idea of lipid-based biomarkers. And that's going to be really important because the technology's not there. Like even if we were to discover a lipid-based biomarker today, the infrastructure doesn't exist in our healthcare systems anywhere where we could mainstream an assay that could use that. So even if we got really strong results today, it would take a while for that to permeate the space to where that could be like a standard test that your doctor orders.
Yep. Fascinating. That is really interesting stuff. So I know you've got some posters that you want to talk about as well, but before you do that, I might jump into some stuff that I was sitting in today, which was looking at risk factors for developing MS, touching on a little bit of risk factors for disease severity and progression as well, but largely around developing MS. And looking at environmental and genetic and epigenetic risk factors. And what I thought was interesting in this session, there was a couple of interesting risk factors that they talked about, but also what I thought was largely interesting in this session was now how we've sort of known that there's a genetic component to MS. We've known that for a really long time. We know that it's not the whole picture. Knowing that, we've known that there must be some environmental risk factors that also play a role. They've sort of been really analysed separately for quite a long time. And now what we're seeing in the presentations today was that there was a lot of interest in trying to work out how those environmental risk factors intersect with the genetic risk factors and trying to see what may work together so that we can work out how those puzzle pieces fit together. I'm not going to get into the specifics of the different genetic markers that they looked at.
Mostly because you're not going to be able to remember it all.
There's, off the top of my head, very good point. But I think they're turning the lights off on us. Let's hope that lasts until the end of the video. But there was a couple of interesting risk factors that I think will be interesting to follow up to see how they develop. One was, you're going to remind me on this, it was weed control exposure.
Weed control exposure,
So essentially exposure to chemicals that are sprayed on weeds to be able to obviously help in the garden and on farms and properties like that. And that they showed from that it was around a certain exposure to that increased your susceptibility of MS by about 3.5 times.
That's really cool. I mean, that's been a theme in the States. That's been a problem for a while with a lot of the agents that they used in the 50s and 60s, like the long-term health effects from those kind of like, they're basically a derivative of like Agent Orange. Yeah. So that's fascinating.
And so they then went and broke down, they looked at some common components in that and tried to see how they affected the risk factors, and then how those common components also interacted with certain genes to see if there was any that interacted with genes that had been identified as being risk factors in MS, and they did find that there were certainly some that did have an overlap. So it'll be interesting to see how that story develops. And the other environmental exposure that they looked at was air pollution. which is a story that we've seen a little bit for a while. Do you remember off the top of your head what? I think it was closer to 1.5 or 2 times. It wasn't as large as the weed exposure control. But that's a story that we've seen a little bit as well, where they've shown stories around how close a person lives to the city, and as further out you get, how that sort of decreases susceptibility around that. So obviously identifying these environmental exposures is of interest, because obviously if we can minimise the risk of people developing MS, that's a positive thing. But also understanding how they're interacting with these genes and the mechanisms behind that is obviously going to help us get a better understanding of what leads to the development of MS, and again, that's going to give us a lot of insight into the disease in general and how we might be able to target that moving forward. So I found that session really interesting.
I think that's really interesting though, because I think these problems aren't going anywhere, right? Like, air pollution is a problem that I think, the more that we can start identifying the risks that aren't just associated with respiratory disorders, but also with like long-term autoimmune issues, those are things that really should affect public policy. And like industrialized agriculture is a big reason why we even have these kind of more and more aggressive like weed control, interventions. And these are things that from a public policy standpoint, like I think increasingly you're going to start to see how they bleed over into other issues. And I think it's really important that we understand the societal risks so that we can we have to be able to mitigate those risks. It can't just all be medicines.
Well, and the other one in there was some look at smoking and how smoking impacts on epigenetics, so basically how environment impacts on genetics. And that showed a really significant effect, and that effect is actually stays around for quite a while even after you finish smoking. I think it took about 5 years for most people.
About 5 years, yeah.
For people to get their epigenetics back to what it was for people who didn't smoke. And I guess that ties in with a really big campaign that we've had in MS for a while around quitting smoking, because we know that smoking increases susceptibility but also worsens disease progression. But we do know that it doesn't matter at what point a person living with MS stops smoking, they do get benefits from that. So I guess this ties in. I mean, we're seeing this in so many levels now that it's really becoming overwhelming, the message for people living with MS to stop smoking, and really people in general to stop smoking. So that was my session. Travis, I know you went to the poster session as well. There's a couple of things there that you wanted to talk about.
Yeah, this is day 2, and I kept saying I wanted to get down to the posters.
I mean, the poster hall is enormous.
Yes.
I mean, there's
it's also hot yesterday.
Plus posters in there, I would say. So yes, there's a lot to go through.
Yeah.
But what were the ones that caught your eye?
So I want to be really clear that like about 2/3 of the posters I'm not actually qualified to even read. There's a lot of stuff on imaging, which I'm not a physicist. A lot of the like you would either have to have a math or a physics degree to understand a lot of the crazy imaging and diagnostic stuff that was going on. But cool stuff going on there. A lot of it was like statistical analysis of large data populations. I can read the conclusions and tell you the exciting things that they found, but I wasn't, So a lot of what I kind of migrated towards is the biomarkers, the clinical trials, and the mechanisms or new target identification, like new mechanisms we might be able to intervene in disease. So I'm obviously biased by those, but there's a couple, there was 2 posters that I think that I really liked the most. They would have won if I was in charge of like giving them prizes. But there was a bunch of there's a bunch of posters on cladribine that I thought was really fascinating, because it was a lot of posters kind of telling the same story of this, orally available molecule with a really favorable kind of administration profile, like that's not super demanding on the patient, that is getting pretty close to some of the efficacy profiles of these infusion type of treatments. Now it's not as good, but like the fact that it's better than the other orally available molecules out there. I mean, I didn't see like a comprehensive telling, but I think that the 4-year progression or relapse free rate was like 76% with cladribine treatment, and the data gets a little bit iffy beyond that, because I think that the biggest cohort came online 4 years ago, so we don't really have much data beyond 4. But that was, even though it was inferior to, it was getting close to, some of the injectable or infusion-based kind of therapies. The other two posters that I saw that I really, there was a lot of themes that were redundant. BTK, serum GFAP, neurofilament light chain, the CYP1. There was kind of like, whenever there was a poster that had a new idea, I was like, oh, I'm going to look at that. One of them came out of Oregon Health and Sciences University by, and I'm not probably, almost certainly going to butcher this name, but Kanan Yoshihara. And I was immediately hooked by this idea of, he was looking at the lesion, the content of MS demyelinating lesions. And he was highlighting how hyaluronic acid is actually a major, unique component of those lesions. But it's actually not HA that's problematic. There are enzymes, hyaluronidases, I think that was close, that break down that molecule, and the fragments are actually super inhibitory to OPC differentiation. And that's what stops cells from being able to make new myelin. So they actually looked at trying to develop a small molecule, or though I'm getting that confused, but basically they were looking at small molecules that could actually interfere with some of these like small fragments. And it was just a really kind of cool and unique approach I've not ever seen before. And so I'm actually really interested to kind of learn more about that approach, and I made a note to go back. The target's actually CCMIP is what they're making the drug against. It's like the receptor for these, actually no, I think it's the enzyme that actually converts the HA into the small molecules. But novel target. The other one was, and this was actually kind of timely. So Arnon Kara, who's a professor out of Israel, who couldn't make it because of the tragedy that's kind of unfolding in the Middle East. So he had one of his colleagues kind of present the poster for him here. But they're looking to take into a phase 1/2A here in the next couple months a small molecule inhibitor of the bone morphogenic protein, and I'd not ever seen that before. But they had a really good literature history and a really good kind of demonstration of how BMP was kind of important for a lot of the signaling events that lead to poor outcomes, failed remyelination, things of that sort. And the data was really compelling enough that they were able to get kind of the IND equivalent in Israel, and they've successfully licensed that molecule. So there's actually a company that's already coming in and looking at it, and it's just it's funny because something was good enough for somebody to come in and actually pay the money to conduct a clinical trial, but it hasn't become pervasive enough that we hadn't heard of it. Yeah. And so I got a really big kick out of that. Maya Golan, I think, was the first author on that, and so that's another one I'm going to be looking into, and maybe your audience would be interested in kind of looking into themselves.
Yeah. So I think that sums you up really well, because I'm pretty sure you told us there was 2 posters that you wanted to talk about, and you talked about 3.
Checks out.
Very Travis. So actually the last talk I want to focus on before we finish up was actually a talk from Dr. Jennifer Massey, who is based in Sydney, so coming out of Australia, which was really touching on 2 very topical issues in MS that we talk about a lot, which was AHSCT, so stem cell transplants, but looking at that in relation to EBV. And so what's happening at the moment in terms of the transplants that are happening is that they're not getting sorted at all. So it used to be a process where they would sort and try and remove some cells that they thought weren't necessary, but that doesn't happen anymore because they thought there weren't necessarily benefits and maybe more risks to it. But what she was interested in looking at then was, are we putting back in cells that are EBV positive? And so what she saw is that yes, post-transplant, these individuals did have memory B cells that were EBV positive, so contained EBV. And what they then wanted to go on and see was, do we then get an immune response generated against EBV due to that? They showed that there were CD8 T cells, so these are just cells that are in the immune system that attack largely virally infected cells. They did expand in these individuals, and actually what they saw was that there was an increased diversity of EBV-specific targets post-transplant to what there was beforehand. So this is quite an interesting finding, and considering what we're starting to think about EBV, there's a thought of, could this potentially be a negative consequence of this stem cell transplant? But interestingly, what they saw was that there was absolutely no clinical relevance to this at all. So out to 2 years that they looked at in this study, all of the patients in that had complete clinically stable remission. There were no changes on imaging, no disease progression at all. So it's an interesting thing as we start to look at EBV trials in people living with MS, where they're looking at what happens if we clear EBV. As we're looking at that, seeing that this immune sort of presence that's there doesn't seem to make an impact on the clinical relevance, it'll give us some insight. So it'll be interesting to see how that story develops.
You know, I'd be interested to see what the different antigen recognition motifs of those antibodies were. Like, is there something about somebody who doesn't have MS or like, you know, healthier cells versus, you know, an MS patient? Like, are we actually recognizing different epitopes?
Yeah.
Is that protected?
Yeah, I think that opens up a whole heap of questions of looking at that sort of stuff post-transplant and AHSCT as well, to see is this something that determines whether or not someone has remission for a really long time versus people who, you know, have a little bit of benefit and then start to progress again. So a lot of interesting stuff. I think we were hoping to, again, keep it brief. We've definitely failed at that again, but hopefully...
He says that, but he keeps asking me back.
I do. Hopefully that's been an interesting insight. The Day 2 ECTRIMS podcast will also be released, where I interviewed Claire Walton, who's the head of research at MS Society UK, so give that a listen as well. She had a lot of insights from her experience at the conference today. We will be back tomorrow to do our Day 3 summary. Once again, thank you to Joe and Travis for joining me. If you do have any questions, post them below the video and we'll be happy to answer as quickly as we can. Thanks, everyone.